Hello everyone, my name is Lamisha Shah, and today I'll be presenting my poster on Group B Streptococcus adhesion to vaginal and bladder epithelial cells in diabetic conditions. I worked with my research mentor Vicky Mercado and Dr. Catherine Patros from the Immunology and Microbiology Department of Baylor College of Medicine. And so to give you a little bit of background on Group B Streptococcus and the study, I'm going to also just interchangeably use Group B Streptococcus with the acronym GBS going forward. And yeah, let's get into it. So the rates of invasive Group B Streptococcus, obesity, diabetes have increased in US adults. Studies have supported that obesity and diabetes are associated with increased risk of GBS infection. GBS is a gram-positive coccus bacteria which grows in chains of diplococci. GBS can cause neonatal and maternal infection during pregnancy and postpartum, endangering both the mother and fetus. Those highest at risk for GBS infection are pregnant women with gestational diabetes. Pregnancy-associated GBS is established as a cause of neonatal morbidity and mortality. And so this study's research focuses on GBS adhesion to vaginal and bladder epithelial cells in diabetic conditions of an in vitro model to understand the correlation of gestational diabetes with GBS infection. And to go a bit into the methods and our experimental design, first we had our research question, which was, do diabetic conditions alter GBS adhesion to vaginal VK2 epithelial cells and bladder HTB9 epithelial cells? We hypothesized that diabetic conditions increase GBS adhesion to bladder and vaginal epithelial cells. And we broke this up into two experiments. First experiment, we looked into GBS adhesion to VK2 vaginal epithelial cells, and we started with a GBS subculture using two strains, one of them which was COH1, the other was 17B. And what we did, we wanted to create these increased glucose diabetic conditions within the subculture of the bacteria. So first, in one of them, we use the control as 0.2% glucose, which would be like the normal amount of glucose we would add. And in the diabetic condition ones, we added 0.4% glucose, so double the amount of the normal glucose that would be there. And then infected with the MOI of 10 to VK2 vaginal epithelial cells, so we added 100 microliters of the bacteria diluted in a 1:10 ratio. So we also wanted to mimic the diabetic conditions not just in the GBS subculture, but also in the host cell. So for VK2, we also had some with the normal amount of glucose added of 5 millimolar, and then we double the amount of 10 millimolar of glucose for the increased glucose diabetic condition mimicked one. And then we washed and lysed the cells, plated 25 microliters of the sample of the adherent bacteria on agar. So on our agar plate, we had seen the GBS adhere to the VK2 cells and counted the colony forming units and calculated the percentage of the bacteria recovered. And then in our second experiment, which was a very similar design to the first experiment, basically except this one, we just looked at GBS's adhesion to HTB9 bladder epithelial cells, and then we added an extra strain of GBS to the subculture, this time being the NCTC strain, just to see if that could change or vary in the data, and how GBS might adhere to that bladder epithelial cells. And we followed the same procedure, and like counted the percentage of bacteria recovered in adhesion. And this resulted in some adhesion to the BK2 cells and HTB9 cells, but we wanted to look further into that with the data. So here we have a graph where we are showing GBS's adhesion to the VK2 vaginal cells with and without the extra glucose conditions. On this axis you'll see the percentage of adhesion, and on the bottom you'll see the types of strains we use. We didn't really see any trends in either strains, so like with the glucose and without the glucose, with the glucose in the GBS and the glucose in the extra glucose in the host cells. So we also looked at the data for the HTB9 bladder epithelial cells with adherence to GBS, and there was a lot of variability in the data with our replicate data, and no like patterns or trends, especially with the range of variability. So we were in a situation where now we're analyzing our results, and we concluded that from the study, the increased glucose conditions in the host cell and pathogen did not consistently increase group B Streptococcus colonization in our in vitro model of the VK2 vaginal epithelial cells, nor the HTB9 bladder epithelial cells. So it did not support our hypothesis for the in vitro model. However, the differences in the GBS colonization may be associated with some other factors, and the findings of adhesion to our host cells were inconsistent, but there are other variables that we haven't captured in the study. So in our next steps, we're thinking about an experiment in an in vivo model using a mouse model, and thinking about other aspects of GBS that may be affected by glucose other than adhesion, such as what are like other ways high glucose can impact GBS in terms of growth, metabolic process of GBS, and virulence factors. And that basically wraps up this study and the paper right now, and I wanted to give acknowledgement to my research mentor, Vicky Mercado, and Dr. Catherine Patras, the Patras Lab, Baylor College of Medicine, and the American Physician Scientists Association. And thank you.